move now to the second speaker. That is Caroline Lam from the Imperial College. She will go on describing formal tools for synthetic biology. Caroline, please.
Good afternoon. My name is Caroline Lam. I'm representing two institutes, the Imperial College London and in UK, and Helmholtz Center for Infection Research in Germany. And not to forget, we have a third collaborator, CSIC from Spain. Today my topic is about formal design tools for synthetic biology. I would like to elaborate a bit more to particular, I'm going to discuss engineering the building blocks of an archetypal genetic circuit in Pseudomonas putida, which is a strain that can degrade aromatics, which is a compound that causing a lot of pollution problems. A rough outline. First of all, I will discuss some background why we want to use mathematical models to simulate Pseudomonas putida, and then about the bacterial strain, and then next about the plasmid that is responsible for this aromatic degradation, and then the method of the circuit modeling. And this project is still ongoing, but I have some interesting initial results to show you, and finally the summary. So first of all, this is a phylogenetic tree showing from Verza et al. In 1990, showing how they perceive the evolution of life based on RNA data. And this is, this is a electron microscopy graph of a rod-shaped bacteria 2 billion years old from Ontario, from Schaaf et al. In 1960s. And this is a picture of E. coli from a few years ago. I just want to show you the bacteria, they evolved based on the historical environment. And it makes people think, what if the historical change was different? Will it give some bacteria that behave differently, which, for example, degrade certain substrate more effectively, or have some other properties? Just want to highlight the Pseudomonas putida strain that we are looking at belongs to this gamma proteobacteria. So this is the genome of Pseudomonas putida. This is a picture of it from Harwood et al. It's not working. Okay. And this strain, it can degrade quite a few aromatic compounds. This is just two examples, toluene and xylene. Just now, Professor Philip Wright has already mentioned the difference between system biology and synthetic biology, and he has explained why we want to use synthetic biology, so I'm not going to discuss any more about that. However, I want to emphasize we have got so much data now from synthetic systems biology, all the DNA information, the protein information, and the metabolite information. We believe it is important to translate all this information into mathematical languages, which can help us test hypotheses and also design some more future informative experiments. As we all know, cell culture experiments is very time-consuming and expensive to perform. So once we got some previous data, we don't want to always use only experiments to test any future questions. If we can learn something from any past existing data. And also, it's very often that all the experimental data out there, they don't use exactly the same cell system, for example, not the same bacteria, not the same mammalian system, and they don't always stick to the same conditions and triggering factors. If we can collect different the results from different experiments and store that data in terms of mathematical formulas, parameter values, then it will be easier for us to compare to see if there is a common pattern, or to see if some information is missing in the particular cells that we are looking at. Can we borrow it from other results that has been done before? So this is our Pseudomonas putida. If we look at it closer, we can see this is the chromosome containing the DNA, and this is the ribosome. And this one is the plasmid that contains the all the genetic material to degrade aromatic compound. I'll give you an example. This plasmid, it contains two clusters or two operons. That is, first of all, the upper pathway, after three reactions, it would degrade silane into methyl benzoate. And then another meta pathway, it would end up producing pyruvate and acetaldehyde. And pyruvate, it would enter the TCA cycle to produce energy. This is the tow plasmid, which contains 4 promoters, PU, PM, PS, and PR. And it can be, and different parts of this plasmid can be activated by different aromatic compounds. For example, if it has silane here, it will activate PR, the protein translating the gene translating sour protein. And here, for the meta pathway, we need 3-methyl benzoate or a similar derivative to activate it. If we imagine these different parts of the plasmid as logic gates, because they require different inputs, and sometimes they need all of them to activate it. Sometimes they have backward inhibition, and try to represent them as logic gates, like AND gates here, and then OR gates here, etc. We could produce something like that. This one is a logical representation involving only binary variables. The ovals are the input. For example, here we have silane, HU. These are sigma 70, sigma S, sigma 54, some housekeeping factors. And the rectangles are the output. This one is the upper operon, this is the meta operon. And here we have different AND gates and OR gates representing how they need to be combined in order to produce the activation or inactivation required for this plasmid to be functional. Here, red means they are off. Red means they are not off. Green means they are on. This yellow is simply to highlight this is a product inhibition system. For example, if I activate sigma 70, IHF sigma 54 link sigma S, here this OR gate is turned on, but this is still off because we still need this SARA protein. This one is on because we have already sigma 70. And this one is on, the rest is not functional. But if I also add 3-methyl benzoate, then I will have this SA protein activated and also this meta operon. This one is a binary representation of the tow plasmid, which we find to be insufficient to represent the real dynamics of the system. So we try to improve it by converting this binary table for AND gate and OR gate, for example, to a continuous system which still behaves like an AND gate or OR gate. However, now it has a continuous connection. For example, this is 0 to 2 in the x and y axis. If we have both of the input being high in numerical value, we will have the output tending to 1, or otherwise they will be near 0. And for an OR gate, now it becomes, if either one of the input here is high, then we will have an output which is closer to 1. And how fast the system would be activated or inactivated continuously would be related to certain parameters that we set. And we can set it to be at any level in between the highest and minimal and lowest value of these equations. So this is how we translate how we improve the such representation into a diagram. Now this is a dynamic representation where all these promoters, PR, PS, PM, and PU, they are now continuous variables and no longer binary. But some of these inputs, because they are housekeeping factors like sigma 32, sigma 38, and sigma 54, not these two. These two, they are complementary. One would be activated when the other is not. Then we would keep the binary features of them. The model is simulated in gPROMS, which is a software created by Professor Pantelidis in Imperial College London. This software is very versatile for model development because it's automated for dynamic system simulation, parameter estimation, and optimization. This screen is showing an example of optimizing different variables by assuming it's piecewise linear and having the time interval extremely small. So it's essentially continuous. Now I want to give an example of part of the whole circuit. This one only involves the PS promoter, and the upstream element would be Winian aromatic compound, M-silene, and this SAR protein would activate PS if both H2 and sigma 54 are present. And then this would activate SAR-Si protein into SAR-SA. Here I means inactivated, A means activated. And then when 3-beta-benzoate is present, then we can have SAR-SA, which would also further activate downstream element. And in particular, I will show some simulation results for the PS and SAR-S protein. This is an example of how we model PS promoter. This is the transcription rate of the PS promoter. Here we have a basal activity because experimentally it's observed that even when none of these factors are present, it still has a very low transcription rate. However, it's not certain whether it is only from sigma 70 or other housekeeping factors. But in terms of mathematical representation, it's sufficient to use a constant to represent that. And then we have a whole term representing HU, SAR-RA. Here, sigma 54 is a housekeeping factor, so we can assume it is always present. And as an mRNA, it degrades, and the normal half-life of mRNA is the order of a few minutes. And the last term represents a toxic level inhibition behavior, which is observed in a lot of bacterial cell cultures. As we all know, aromatic compounds can be toxic when they are very concentrated. So we have a threshold that we won't let the cell be killed by too high aromatic concentration. This too is the equation for SAR-S protein for the inactivated state and the activated state, which is function of PS and the 3-methyl-benzoate, and also its own concentration and the activated molecule concentration. For the activated SAR-S protein, it only has the last 3 elements because it's not dependent on the PS. This is a comparison of a literature result from Kim et al., where they compare the total plasmid PS promoter activity when they put certain silane in the cell culture and then wait after 2 hours to see how much more PS activity has increased compared to the basal level. The black circle is the experimental results, and the dotted line is our simulated value. And we are happy that we are able to capture the behavior. It's interesting to highlight that for this culture, the toxic inhibition level is around 2 millimolar, which is not very high. That also means it's important if we can engineer the reaction system to make sure we don't kill the cell while we want to remove the harmful molecules in whatever industrial waste product that we want to treat or handle. The model involves quite a lot of parameters compared to what we can measure. To solve this problem, our Imperial College side has developed an algorithm to estimate with the relative importance of all the model parameters, so that for those that are not significant for the model output, we can fix them at the nominal value. And for those parameters that is really determining how the system behaves, then we can directly estimate it from the limited number of experimental data. I'm going to show you some experimental results from data that we obtained from literature. For some, we got it from Putida string, but it's from a wide range of aromatic substrates. and also some other is from toad, but it's in E. coli. This one is a batch culture. This one is a batch culture while it's feeding M-xylene and also succinate. The reason that they need to feed succinate is because if they only feed M-xylene, the cell actually don't grow a lot, or even they die initially. So they have to dope the system with a succinate which directly feed in the TCA cycle. And here is the biomass concentration. This also highlighted because for this example, we can see we are feeding it 15 millimolar of succinate in order to degrade 1 millimolar of M-xylene, which as an engineering point of view is a very much waste of resources. If we can use engineering tools to optimize this system so that it can consume less energy and resources to remove the same amount of undesirable molecules, then we will have a more efficient system which is cheaper, more efficient, and more desirable. This is the one that we tested with 3 additions of substrates. Here I only show M-xylene because the succinate level is so high that it would just squash this line into just a straight line. Here we test it by feeding the same amount of M-xylene as pulses every 2 hours after it has been exhausted. And the system is behaving very properly. We have slightly more biomass, but we can see the M-xylene concentration does not increase to the same height in the 3 consecutive pulses because we have more biomass to consume it. So each time it disappears faster. And the model is doing it properly. This is to show the PS transcription activity and the SARS-I protein concentration for the same batch system as the previous slide. The y-axis is only relative activity and relative concentration for both variable, because when the mRNA transcription rate is analyzed and also the SARS protein is often a qualitative data. They compare it with the system when it's not activated or when it's in a control stage. So we have to convert it into relative activity as well. But still, it's good enough that the model is able to let us know the relative increase of the system. And we can see the two, if you remember, PS is activating the production of SARS protein, but the dynamics of these two variables are quite different. This one stays high in the beginning, and then gradually the free addition of xylene triggers it again, but it's decreasing. This one, it decreases a lot more rapidly, but then these 3 peaks is different from the decreasing trend. I would say it's quite random. We also tested for continuous cultures using the same set of parameters. We are not trying to play around with the parameters when the system is not behaving. We stick to the same results and see if it gives something that makes sense compared to what is reported in the literature. And we are happy to see it's doing very well. Here we have a continuous system where we keep feeding M-xylene at a very low concentration. Again, we try to stick lower than 2 millimolar, except here, but then this one, it comes back down because the cells keep digesting the substrate. And then I step up the M-xylene concentration for 3 times. As we see these 3 mountains, and this is the biomass concentration. Here we see it decrease a little bit because it's a continuous system where the cells leave the reactor. So when, for example, there is no more M-xylene and also succinate, then the cell doesn't grow that fast, and so the dilution rate flushes the cells away. And here is the results for the PS transcription rate and the SARS-I protein. I just want to highlight here the PS activation rate is more or less constant for the 3 different stages, but it's increasing for the SARS-I, which shows it's a truly dynamic system when one responds to the changes in the M-xylene dose, but the other doesn't. Lastly, a summary of what I have shown you. we have a plasmid in Putidar, which is degrading toxic aromatic compound. And to understand this system more systematically and to better apply a lot of existing well-established chemical engineering control tools to optimize this system, we convert it into a hybrid model that contains continuous variables for all the important promoters represented by the AND gates here, and the proteins, SAR-RI, SAR-SI, etc., and the operons. But for certain housekeeping factors, we still keep it binary because they can be assumed constant. We seek to have a mathematical tool that can advise us later on which part of this circuit is the best candidates to be either reconnected or completely changed to make the system more efficient. For example, consume less succinate but degrade the same amount of M-styrene or other aromatics, or for some aromatics that it does not degrade at the moment. For example, some chloro or fluorinated aromatic compound, which is some of the most toxic aromatic compounds that we can think about. Can we make it degrade that and efficiently so that we can have a better plasmid and a better strain for industrial application in the future? At the end, I need to acknowledge all these people and also the Probacter Sensitimo project funded by the European Commission and BPSRC in UK and the EU. And also all these people from Imperial College London, from Helmholtz Center of Infection Research, and from Spanish Center of Biotechnology in Spain. Any questions?
The talk is open for questions. Well, I have a very direct one. So you showed quite a number of parameters. You know, I think when you have this here, you showed quite a number of parameters here. And I mean, in reality, one can develop very complicated equations to describe dynamically a process. But usually, the bottleneck is not so much on the development itself, but is on finding the right parameters and how to measure them. So if you have so many parameters, how can you make sure that the error on determining parameters does not overcome the total result of your simulations? Do you have any
that's a very good question. Usually a lot of models, they go too details into the system, and the level of complexity of the model is dependent on what I want to achieve from the system optimization using mathematical equations. In this case, we simply want to reshuffle all these units in this plasmid. That's why we are not modeling the DNA level yet. But if you want to, there's no problem for that. Already for this kind of, for this not so big network, the number of parameters involved is already 2 to 3 times the number of measurable variables. We have developed a global scientific analysis technique that would analyze within the low and upper bound range of the variation, numerical variation of each parameters, whether it would really affect the model prediction output. For example, if in this equation we have 1, 2, 3, 4, 5, 6, 8 parameters, but we are only measuring one variable. So it's 1 to 8, which is even more than 1 to 3. For example, this is the Hill function coefficient, which in terms of experimentation, there has been a lot of models using this type of equations. And we know it can never be less than 1, and it's rarely higher than 10. Then we take this range and then put this into the parameter scientific analysis, analyzing it globally for this entire nonlinear model to see whether this would affect the output. For example, it would affect the SAR-SI protein activation at a certain time slide in a batch culture or in a chemostat culture. Will it cause any significant effect? For this one, for example, we found it would not matter if the order of magnitude is between 1 to 3. But if it's higher than that, if it varies more than threefold, then it could be a problem. Then it will be something that we need to do more experiment or have more understanding of the biology to see if we can narrow down the range of variation.
Okay, just a question. Have you modeled the metabolic effect of this plasmid? on
the
this?
metabolic effect? You
Yeah.
mean the metabolic degradation pathway?
No, I mean for the enzymes, because this plasmid has
We
some
are also
enzymes.
modeling the enzyme, but I'm not showing this because we haven't got the data. We are doing experiments, and there isn't any detailed literature data out there yet, so I couldn't show in this early stage, but we are indeed working on that. That's the whole idea. Without the enzyme, we cannot degrade the aromatics.
Alfonso?
And here you seem to you pick up a choice of logic gates.
Yeah.
And although you use analogic, so you fit with an analogic function, but how dependent is your predictions on the choice of the digital behavior? Because, for instance, it may happen that the real promoter is between and or. Then it's not captured by that, and then if you fit against this digital behavior, then you may just get something that is different.
Let me get the question right. Are you asking what if it is not an AND gate? What if it's an AND gate plus an OR gate?
Or something...
Something more complex.
Yeah.
The reason that we choose an AND gate is because from all the existing literature data, they are only suggesting when all three are present, then they see an activation.
But yeah, but that's poor data. So I think so.
Yes, I'll agree. You mean should we look more into the experimental evidence?
Yes, so sometimes constraining that, then it's a lot of constraints. So then I mean that
I totally agree with you. In fact, I haven't shown here, but there are other parts of the circuit which we found contradicting literature data. For example, the PM promoters, some said when it's activated by quite a few upstream molecules. Some said when all the upstream activating molecules are there, it will be activated even more, but some said it's the same. For us, we would test it with our results to see if we see that. And for example, for PS, it's even worse. Some said there is a background activation from sigma 70. Some said it's not sigma 70. Some said there is a low level of automatic activation when there's nothing out there. And we have tested it with a few different models, but we find it does not affect the model outcome so far with anything that is important for the aromatic degradation. So we stick to what is useful in terms of engineering. And for this uncertainty here, we would have a library of different equations, which we would keep testing until we decide it's no longer necessary to do that, and then we would just pick the most confident one. I'll agree there are a lot of uncertainty, but at the same time, I see the opportunity to look at what is the most important concern, and to answer those questions first. For those that are less certain, less clear, but if it can have a lower importance of the system output, we will keep that for further scientific research to understand it better. But we don't want to kill the present application.
All right. Just one short question to this, if I so actually, the system you showed is a lot bigger than this, and you already have so many parameters here. Even using your reduction parameter reduction modules, still in the end you'll end up with certain error. And if you combine the various modules to each other, in how far are, let's say, the simulations representative of a system? That how is the error propagation in each of these circuits, and when you combine them together? Is there not a limitation in size of how far you can tackle these systems? Because it looks already complicated enough for a very small one. So if you try to even describe just one plasmid, it doesn't even seem that it's possible somehow. Or
I do believe that could be a limitation of how far we can scale this up with so much uncertainty going on. We haven't tested it yet, but there's no reason why we shouldn't. But everything has a starting point. So I would say let's start from a smaller system, something manageable, and once we get that system working, then we can try to test it on something bigger. But at the same time, this cannot be. This can never be in a in a scale that is the size of a cell, because in that system we have the order of thousands of genes, and if we still only know less than 10% of the variable, I don't think it would really work, unless we do more experimentation to reduce this uncertainty ratio to, let's say, 1 to 2 or something even less.
All right, thank you.